Hello, my name is Marisa Saloki. I'm from Creighton University School of Medicine, and I'm going to present our work entitled Characterization of Quinoxaline Derivatives for Protection Against Iatrogenically Induced Hearing Loss. I am part of the Translational Hearing Center at Creighton University, and the goal of the center is to identify therapeutic compounds that will protect against different types of hearing loss. The pipeline that we have at the center is the modification of different small molecules by medicinal chemistry, followed by screening and identification of lead compounds using zebrafish as a screening model, and then those lead compounds can be further characterized for potency, efficacy, and mode of action in zebrafish, and then the corroboration also of these lead compounds in a different model for ototoxicity, such as mouse cochlear explant. And then finally, at the end of the pipeline, we're interested in identifying the molecular target of these compounds and move the compound forward into mouse studies that are more relevant models for ototoxicity. Quinoxaline was our original molecule. We found that this molecule has a potential as a therapeutic against hearing loss, but we wanted to improve the protective effect of quinoxaline against aminoglycosides. So we synthesized a library of 70 different derivatives, and we found that after the screening, we found that QX28, that has a carboxylic acid in position number 5, was the one that showed the best efficacy and the best potency against aminoglycosides. Here is the standard dose response curves showing in red QX28 and in green our original molecule quinoxaline, and in a system where fish were incubated with neomycin as the ototoxic agent, and we see that we were able to see that QX28 has a better potency and a better efficacy than quinoxaline itself. Similar results were observed with gentamicin, a different aminoglycoside that is commonly used in zebrafish models. We observed that in the presence of QX28, the drug that was labeled with Texas Red, gentamicin or neomycin, the drug was able to get into the hair cells, suggesting that the protective effect of QX28 is intracellular and is not due to the blockage of the mechanotransduction channels. And finally, since quinoxaline is known to inhibit the IKK activity, we wanted to test if also QX28 is involved in the NF-kappa B pathway as quinoxaline. And we use a reporter zebrafish line that expresses GSP under the regulation of NF-kappa B binding sites. And we observe that in the presence of the ototoxins alone, we observe an increase in the fluorescent intensity of GFP, but when the animals were co-incubated with QX28, there was a reduction in the fluorescent intensity, suggesting that QX28 is inhibiting an NF-kappa B pathway, as it shows here. So as a summary, we want to show that in the presence of the toxin alone, the NF-kappa B pathway is being activated in the supporting cells of the that surrounds the hair cells in the neuromass, and that results in hair cell death. But when we co-incubate the animals with a low dose of QX28, the NF-kappa B pathway in supporting cells is not being activated by the ototoxin, and then we observe protection in those neuromasts. So as part of the work, other authors that were involved were Dr. Shan Suo, Dr. Santanu Hati, Shen-Han Zhu, William Hausman, Hugh San Liu, and Dr. David He, and this is our support. Thank you very much.